Yeah, like Erin said, my study is on the Cryoconite Holes on Root Glacier in Alaska. I looked at dissolved organic carbon and bacterial abundance. Before I start, I just want to give some acknowledgments. Firstly, to my awesome reader, Erin, and also to Jenna Nelson, who a lot of you know, for all her help with my research design. I'd also like to acknowledge that within the lands of the Wrangell St. Elias National Park and Preserve, there are 4 Indigenous groups that have ties to that land. And of the 4, the Atna is they historically reside in the interior of the park where the glacier is located. So glaciers consist of many complex systems. Of these systems, there are these small water-filled holes known as cryoconite holes. So I thought a good place to start would be to explain to you what a cryoconite hole actually is. The diagram or the figure on the left shows just a basic diagram of it, but a cryoconite hole is formed when sediment from an outside source lands on the glacier and it changes the albedo of the snow or changes the temperature. So the sediment lands on the glacier, it's darker than the snow, and so it's warmer, and so these little pools form. And within these pools, there can be sediment, like I said, microbial communities, and also just plant matter. The cryoconite holes exist on most of the world's glaciers and their ice sheets. Globally, they can cover estimated 4 to 6% of all glaciated surfaces. Dissolved organic carbon, or DOC, we're going to talk about that a little bit. So I just wanted to define that for you a bit. It can be found in cryoconite holes and has been found in many studies all over the world in cryoconite holes. And it's basic nutrient that supports the growth of microorganisms. The reason why I am interested in cryoconite holes and why they first interested me is because with glacial melt becoming more rapid all over the world at different rates, I was curious as to what kind of inputs would be going into our downstream ecosystems. My key research questions for this study was, is there respiration occurring in the cryoconite holes on Root Glacier? Are there bacteria present in these holes? What is the dissolved organic carbon of the selected holes on the glacier? And does the physicality of the cryoconite holes impact bacterial abundance, or the DOC? My study was conducted, as I said, on Root Glacier in Alaska. It is in the town of Kennecott. My fieldwork was on May 19th through the 22nd. I looked at bacterial abundance and DOC concentration in a total of 15 selected holes. I also collected physicality data within those holes. I looked at respiration rate, and I also did 5-meter plots to look at within those areas. These are just a few pictures to kind of give you guys an idea of what the terrain looked like. The upper right corner are a cluster of cryoconite holes. Below that is like a little glacier lake that existed throughout the glacier. There was a bunch of them. And you can kind of see within that picture, there's valleys. And then the middle picture shows the glacial till to the left, and then kind of the undulating terminus of the glacier. And then all the equipment, as Erin said, that I had to carry. I did have help. Thank you, Sage. Photosynthesis and respiration rate. That is the first study that I did on the first day. I collected glacial water from the cryoconite holes from a total of 13 holes. I filled 15 BOD bottles with that glacial water to use Winkler titrations to look at the change of oxygen over time. 3 of those bottles, I measured the oxygen concentration immediately. And then 6 of those bottles, I put 2 to 4 millimeters of sediment in, and the other 6 I left with just the glacial water. Of those 6, 3 I've covered with aluminum foil and left in the dark. So 6 bottles with sediment, 3 of those were covered with aluminum foil, left in the dark. 6 without sediment, 3 of them covered with aluminum foil left in the dark, and those were incubated for 24 plus or minus 2 hours. And then the oxygen concentration was measured again. I found that the respiration rate was a lot higher in the bottles with sediment. As you can see from table 1, the bottles with sediment have significantly more respiration rate than the bottles without sediment. This could potentially show that the sediment provides a substrate for activity, bacterial activity and growth. The next collection I did was for dissolved organic carbon. I collected water from 15 random holes, and in those holes, sorry, the range of dissolved organic carbon I found to be 0.08 to 5.83 milligrams per liter. Just to give some sort of context to how much that is, the average DOC concentration in our global rivers is 10.4 milligrams per liter. The amount found in the holes is obviously less than that number. Potentially a reason for that is you guys can see to the right, the cryoconite hole water is very, very clear. There's sediment at the bottom, but the water itself is basically crystal clear. And river systems obviously have a lot more input into them. So that could be an explanation for why the concentrations are lower. Also, if you look at the undisturbed watersheds in the world, they have a base flow concentration with a range of 1 to 20 milligrams per liter. So the dissolved organic carbon found in the holes is within that range. I brought up how clear the water is because I wanted to tie that back into the respiration rate I talked about earlier and how the sediment had so much more going on. And I think that the dissolved organic carbon numbers really speaks to the crystal clearness and the lack of sediment within the water column itself. These two figures show the 15 cryoconite holes that I measured. You can see that the water is really blue and there is sediment on the bottom. Some of them are a lot thicker than others. The figure 4 on the right shows the sediment after it's been dried out. So there's really not a lot of sediment within these holes. To better show that, I made this diagram. The average depth of holes for the 15 that I measured is 174 millimeters, where the average depth of the sediment was 2 to 4 millimeters. So there's quite a difference in the amount of sediment versus the amount of water. It's mostly just water. So there's not a lot going on sediment-wise in the holes that I measured. The next thing I looked at is bacterial abundance. I filtered the cryoconite hole water through a black polycarbonate filter and then used the epifluorescent microscope to do bacterial cell counts. I found that the bacterial abundance in the holes ranged from 73.4 to 241.8 cells per milliliter. There's several studies done all over the world's glaciers on bacteria in cryoconite holes. The ones that I looked at also used the same technique, and they were in Antarctica and the Himalayas. And their bacterial count was a range of 700 to 95,700 cells per milliliter. So obviously substantially more bacteria were found in those holes than the ones that I looked at. There could be several reasons for that. There are different places. The holes that I'm looking at is mostly glacial till. The sediment is mostly glacial till. Whereas the Himalayas, it could be there's more plant matter in the area. Also, seasonality can play a big role on the amount of bacteria that will be found. I did my study in May, so the beginning of the melt season. So the holes are just opening up. It hasn't been very sunny. It's Alaska, so there's not a lot of sun yet. Whereas the study done in Antarctica and the Himalayas was done in September. So they had a whole summer season of sun, and each of the holes is its own little ecosystem. There's no input, there's no output or input. So, the whole summer it's a lot of time and sunshine for things to happen. So that could be a potentially a reason why my bacterial abundance was so low. There are several studies done just to tie back into the sediment that show a difference in the water column bacteria versus the bacterial abundance in the water column versus the bacterial abundance in the sediment. And within those studies, the bacterial abundance in the sediment is much more. I didn't find any correlation between any of the physicality data that I collected, pH, temperature, size of the hole, depth of the sediment. There's no correlation between any of that and bacterial abundance and any of that or in DOC. However, I did find a negative correlation between bacterial abundance and the DOC. This reflected other studies that showed an increase in microbial abundance with a decline in DOC values. My p-value was 0.0035, and like I said, it was I had an R-square that showed a negative correlation. This could suggest microbial activity is using the DOC within the water columns. I only had 11 data points. I lost 4 vials in transit. And as you can see, they're kind of clustered. So more data points would be ideal for that. The next thing that I looked at was I did a 5-meter radius plots. I did 3 of them, just to look at the density of the holes on the glacier. I found the number of holes to be from 106 to 184 holes within each of these circles. I then used the minor axis and the major axis of each of the 15 cryoconite holes to determine an average radius, and then use that number to determine an average surface area of each of the cryoconite holes that I looked at. I found that number to be 51.95 centimeters squared. And then with that number, I calculated the surface area that would potentially be covered in the 5-meter radius plots, which was from the lower number of 0.7 to 1.2% of the total hole. The biggest takeaway or the most interesting thing I found within this study was that the respiration rate in the BOD bottles, the ones just containing water, didn't have almost no respiration rate, while the ones that had the sediment had such a larger amount of respiration occurring, which means there's stuff going on in the sediment. That really helped me understand, or at least begin to understand, why my bacteria abundance and my DOC levels were low. In the future, I think it would be important to examine the bacterial abundance in the sediment itself and not just the water column. As I talked about earlier, another thing that was interesting was that the correlation between the DOC and the bacteria abundance. More data would be ideal to really look and understand that correlation and that relationship. Finally, the hole density being up to 1.2% of the glaciated surface, I think really highlights how much water is in these holes and how much is potentially available to our watershed. In the next 80 years, I think we all know we're projected to lose about 33% of all of our glacial mass. There are a lot of studies being done more recently. Some of them suggest that the release of DOC from mountain glaciers could potentially be 1.4 times higher than what's currently estimated. It's a pretty big difference, and I think that glacial systems should be included in hydrological models moving forward. And yeah, I guess the final takeaway would be that future research on the implication of cryoconite hole sediment on downstream ecosystems should be and needs to be studied. That's all. Thank you.